Hello everybody, welcome to all. I am Dr. M. Armugam, Assistant Professor and Head, PG and Research Department of Botany, JJ College of Arts and Science, Autonomous, Pudukkottai, Tamil Nadu, India. In this video, just we are going to see about the Golgi apparatus or Golgi bodies. An Italian, a neurologist called Campello Golgi in the year 1873 who discovered the Golgi apparatus. By reporting the existence of such organelle inside a cell, he raised a storm of controversy known as Golgi controversy. The Golgi received the highest honor and awards in the recognition of his work. He shared the Nobel Prize in the year 1906 with Santiago Roman y Cajal for this work and the structure of this nervous system. And this is illustration of the Golgi body. It is found in the cytoplasm, most of the eukaryotic cells, and absent in the prokaryotic cells. In mammalian, that is RBC and sperm cells also, and bryophytes also, it is not available. And ranges between 1 to several with a cell. In a plant cell, several means the Golgi complex or dictylosome also called as. Also called due to their high lipid content, Golgi apparatus also called as lipochondria. Since originally these were known to be network, they are called as dictylosome. So the dictylosomes is the name is derived due to the presence of net-like structure. As known as a Golgi complex, the Golgisomes or Golgi body or Golgi material or Golgi membranes also, etc. So these are the names of Golgi body. The Golgi apparatus of a plant or lower invertebrates is usually referred to as Golgi body or dictylosomes. This is a structure of Golgi body. It has a trans phase as well as another phase is called cis phase. This is a cis phase, this is a cis phase, and this is a trans phase. And it has a sister name. These are the sister name. This is a lumen, and this is a incoming transport of vesicles. This is secretory vesicles. There are two vesicles are there, one the incoming transport vesicles and as well as secretory vesicles. And the occurrence and Golgi apparatus occurred in the cell except in the prokaryotic like mycoplasma, bacteria, and also in some blue-green algae. And eukaryotic cells of certain fungi, sperm cells of bryophytes, and mechispores of red blood cells are also not available of this Golgi apparatus. One animal cell usually containing a single Golgi apparatus, but number vary from cell to cell and from the animal to animal also. Nerve cells, liver cells, and oocytes have multiple Golgi apparatus, but in a single algal cells containing 25,000 Golgi apparatus in a single algal cells. So the appearance as well as the availability of the Golgi apparatus will be varied from one cell to another cell. And the Golgi apparatus is morphologically very similar to both animal as well as plant cells. Its shape and form vary from cell to cell also. Those size and shapes also will be varied from one cell to another cell. Golgi apparatus appears on a complex array of interconnecting tubules, vesicles, as well as the cisternae. The cisternae are flatted sacs. Simplest unit of the Golgi apparatus is called cisternae. This is a membrane bound space in which various material and secretions may occur. A numerous cisternae also associated and appears in the stalk-like structure. The group of cisternae is called as dictylosome, and group of dictylosomes is make up of the Golgi apparatus. Yeah, first second one is tubules, a complex array of associated vesicles and tubules about 30 to 50 nanometer in diameter surrounded the dictylosomes. And vesicles are the three types. One is the transitional vesicles. It forms blebs from the transitional endoplasmic reticulum to Golgi body, and secretory, that is secretion. Secretion of vesicles occurs between the Golgi apparatus to plasma membrane. And third one is a clathrin coated vesicles. It plays a role in a intracellular traffic of membrane and secretory products also within a cell. And biochemical properties of the Golgi apparatus is concerned. One is the glycosyl transferase and thiamine prophosphatase localized in the cisternae. The transferase enzymes are present in the membrane of Golgi apparatus itself. And second one is acid phosphatase present in the GURL. Next one is, and the third one is Golgi apparatus of the animal cells containing the following important enzymes, then like phospholipase, kinase, glycotransferase, and glucotransferase, also oxidoreductase. These are the enzymes which are essentially filled by this Golgi apparatus in animal cells. Now we can see this image. This is a Golgi apparatus. This is a Golgi apparatus. From here we can have these endosperm like lysosomes will also happen, and sometimes the Golgi apparatus will secretory. This is secretory vesicles, and the cisternae will attach with this endoplasmic reticulum also. And origin, how the Golgi apparatus will be originated means the method of origin of Golgi apparatus is de novo formations. Then individual stalks of cisternae may rise from the pre-existing sacs or by divisions or the fragmentation of the existing Golgi apparatus. The membrane of Golgi apparatus originates from the membrane of smooth endoplasmic reticulum also. The cells of dormant seeds of higher plants generally lack of Golgi apparatus also. Here this is a de novo assembly. How this is a Golgi apparatus when give the trans space as well as cis space will be happen after the formation. The template mediated assembly, a Golgi apparatus will be scattered and it is attached with the endoplasmic reticulum. Here the new Golgi apparatus will be formed on the basis of matrix protein enzymes and the endoplasmic reticulum as well as Golgi cyclic protein. And secretions, the Golgi complex plays an important role in the secretion. This is a secretion what we have seen. This is a Golgi, the endoplasmic reticulum will in a endoplasmic reticulum we will have the vesicles. From vesicles it is attached to the Golgi apparatus, and the Golgi apparatus will be secretory vesicles will be produced to secrete the some proteins as well as the secretory proteins. And the functions, the Golgi vesicles are often referred to the traffic police of a cell, because it only allow whether it is secretory proteins will be allowed or not. They plays an key role in certain of many cells proteins as well as membrane continuance. It is a direction of their proper destination also. And the Golgi enzymes added a signal or the tags such as the carbohydrates or a phosphate residues to the certain proteins to direct them in the proper destination also. In plants, the Golgi apparatus is mainly involved in the secretion of materials of cell walls like lipids, glycoprotein, cellulose, hemicellulose as well as lignins. And it involved in the cell plate and cell membrane formation of daughter cells also. These are the functions. It is illustrated by a very good image also. And in animal, the Golgi apparatus is involved in the packing and exocytosis of this one is mucus, that is a glycoprotein, and then is a lactoprotein secretion by the mammalian glands, and third one is secretion of collagens, and fourth one is the formation of melanin and also the other pigments. It is also involved in the formation of cellular organelle like plasma membrane, lysosomes, agrosomes of the spermatozoa and granules of the oocytes. It is also involved in the formation of cellular organelles. That here also we can see this is a Golgi bodies, how it is organized during the maturations, how it is organized. And apoptosis is a very essential action which is in the cell. This is apoptosis is a form of the programmed cell death that occurs in the multicellular organisms. Biochemical events leads to the characteristics of the cell charges as well as the morphology of the death, because in the programmed cell death will be happen in the assist of the Golgi apparatus. These changes include the bleeding, the cell shrinkage, nuclear fragmentation, chromatins, condensation, chromosome DNA fragmentation, and global mRNA decay. These are the metabolisms will happen by the presence of the Golgi apparatus. The average adult human loses between the 50 to 70 billion cells each day due to the apoptosis. For an average human child between the age of 8 to 14, approximately 20 to 30 billion cells die per day only because of the Golgi apparatus, because then only new cells will be generated and the growth will be happen, and cell divisions also will be promoted. And the transport of the Golgi apparatus, the transport from the Golgi takes place by the two ways. One is a constitutive secretory pathway and the regulatory secretory pathway. This is a thing how the secretory pathway will be happen within the Golgi apparatus. And the constitutive secretory pathway is happen. One is the protein are secreted from the cell and continuously regardless of the external signal of the factors. And the second one is the proteins are stored in vesicles in the Golgi as well as move directly to the cell and surface with the fuse with the plasma membrane and release the soluble proteins also. Here like this is we can see the trend phase of Golgi apparatus will be secretory vesicles will be happen. Here we can see this secretory and then how it is formed and how the secretory regulated secretory pathway will be happen. This is the way how the constituent secretory proteins will be happen like that. This is the way how the constituent secretory protein pathway, and this is the way how the regulated secretory pathway happening. And the regulatory secretory pathway or the distinguished regulatory secretory pathway in that which in the specific proteins are secreted in the response to the environmental signaling. And the second one is the proteins are sorted in the trans phase of Golgi network and packed into the secretory vesicles. Their secretory vesicles are usually transitor vesicles and stored in until the specific signals are received, then fuses with the plasma membrane to release the proteins also. This is a selective transport of the protein lysosomes is one is the process of protein shortening in the Golgi by the lysosome through the selective transport system of protein, and the proteins are the modified by the mannose phosphorylation. It is due to the in-cis Golgi networks also. These phosphorylated mannose molecules are specifically recognized by a mannose 6 phosphate and receptors. This only due to the presence of the trans Golgi network. This is the processing of human lysosol proteins happen. How it is happen like this we can see. Thank you very much for patient listening this video. Thank you.